1. Introduction to mitochondria. Cellular powerhouses. Mitochondria are membrane-bound organelles found in most eukaryotic organisms responsible for generating adenosine triphosphate (ATP). Endosymbiotic origins. They are widely believed to have evolved from engulfed ancient aerobic alpha proteobacteria through a symbiotic relationship. Dynamic networks. Rather than static beings, mitochondria constantly fuse and divide in a dynamic network inside living cells. 2. Structure and anatomy. Dual membranes. The organelle is bounded by a smooth outer membrane and a highly folded inner membrane that regulates transport. Cristae architecture. The inner membrane folds are called cristae, which vastly increase the surface area available for chemical reactions. The matrix space. The innermost fluid-filled compartment is the matrix, containing metabolic enzymes, mitochondrial DNA, and ribosomes. 3. Primary function, ATP synthesis, cellular respiration. Mitochondria extract chemical energy from nutrients like glucose through a series of complex metabolic pathways. The citric acid cycle occurring in the matrix, this cycle oxidizes acetyl-CoA to generate NADH and FADH2 electron carriers. Oxidative phosphorylation. Electrons from carriers are passed down the electron transport chain to drive the phosphorylation of ADP into ATP. 4. Secondary functions of mitochondria. Calcium homeostasis. Mitochondria act as critical buffers by rapidly taking up and releasing calcium ions to maintain cellular signaling balance. Apoptosis regulation. They play a central role in programmed cell death by releasing cytochrome C into the cytosol when a cell is damaged. Heme synthesis. The initial and final steps of heme biosynthesis, vital for hemoglobin, take place within the mitochondrial matrix. 5. Tissue distribution. Metabolic demand. Variable abundance. The number of mitochondria per cell varies dramatically depending on the metabolic and energy demands of the specific tissue. High-demand organs. Organs with relentless metabolic workloads contain cells with thousands of mitochondria to sustain continuous operations. Low-demand tissues. In contrast, structural or storage tissues with lower metabolic active rates require far fewer mitochondria per cell. 6. Mitochondria in high-energy organs. The human heart cardiomyocytes are packed with mitochondria, occupying roughly 40% of the cell volume to power lifelong continuous pumping. Skeletal muscle. Muscle fibers, particularly slow-twitch fibers, possess high mitochondrial density to fuel prolonged physical exertion. The brain and neurons. Neurons consume massive amounts of ATP to maintain ion gradients for electrical signaling, relying heavily on synaptic mitochondria. 7. Mitochondria in other major organs. The liver, hepatocytes. Liver cells contain 1,000 to 2,000 mitochondria each to power diverse detoxification and biosynthetic pathways. The kidneys. Renal tubular cells require immense mitochondrial energy to actively reabsorb ions and filter metabolic waste from blood. Mature red blood cells. Erythrocytes possess exactly 0 mitochondria, ensuring they do not consume the oxygen they are tasked with transporting. 8. The mitochondrial genome, mtDNA. Semi-corporate autonomy. Mitochondria possess their own distinct circular genome, reminiscent of their ancient bacterial ancestors. Maternal inheritance. In mammals, mitochondrial DNA is strictly inherited from the mother, as sperm mitochondria are systematically destroyed after fertilization. High mutation rate mitochondrial DNA mutates at a much faster rate than nuclear DNA, due to proximity to reactive oxygen species and limited repair mechanisms. Genetic division. Nucleus versus mitochondria. Mitochondrial contribution. The human mitochondrial genome is remarkably compact, containing only 37 genes that are entirely essential for its function. Nuclear, overwhelming majority. The human cell nucleus contains approximately 20,000 genes, roughly 1,000 to 1,500 of which directly encode proteins destined for the mitochondria. Coordinated expression. Building a functional mitochondrion requires tight genetic cooperation and synchronized expression between nuclear and mitochondrial genomes. Evolutionary gene migration. Endosymbiotic gene transfer. Over evolutionary time, the vast majority of the original endosymbionts genes migrated to the host cell's nucleus. Why keep any genes? Mitochondria retain a handful of genes because the proteins they encode are highly hydrophobic and difficult to transport across dual membranes. Local control. Maintaining certain genes locally allows the mitochondrion to rapidly regulate electron transport chain components on demand. 11. Conservation across species. Functional conservation. The core function of the 37 human mitochondrial genes is universally conserved across almost all multicellular eukaryotes. Genome size consistency. Nearly all animals exhibit a highly streamlined mitochondrial genome containing exactly 13 protein-coding genes, 22 tRNAs, and 2 rRNAs. The animal rule: from fruit flies to blue whales, animal mitochondrial genomes remain remarkably rigid in gene content and compact size. 12. Evolutionary exceptions in other kingdoms. Plant genomic expansion. In stark contrast to animals, plant mitochondrial genomes are massive and highly variable, spanning hundreds of kilobases with non-coding DNA. Fungal diversity. Fungi display intermediate mitochondrial genomes with varied sizes and gene orders, highlighting unique evolutionary trajectories. Protozoan outliers. Some single-celled protozoans like Jacobids retain up to 100 genes in their mitochondrial DNA, the most primitive genomes discovered.